And thank you very much indeed, Yvonne, as well, for a wonderful talk and what's a fascinating insight into this important group of organisms. And goodness. So if anybody, can you please add the questions? I think, Yvonne, we do have you online, I hope.
Yes, I'm here.
Good to see you. Excellent. That was great. Just while we're waiting to see if people get their typing fingers out, so I'd always imagined that chlamydia was something that sort of was an ancient acquisition, perhaps by a theropod or something. You know, you said it's in snakes, which is perhaps not surprising, and then it's evolved, you know, with hosts. But you seem to be suggesting that breaking out of the hosts and moving hosts and perhaps displacing another chlamydia species has happened on multiple occasions.
Yeah, that is a possibility. We're still looking. We're discovering also very much new species, but I think that the tools that we used in earlier times were not so precise. So we always used in the field a lot of serology, and serology cannot discriminate, for example, between Chlamydia abortus and CTC and some of the others. So with the development of new typing methods, I think and of course the whole genome sequencing, we can now characterize the isolates much better, and we can make more, and so we discover new species this way. And you have to remember that chlamydia are obligate intracellular pathogens, which were really difficult to culture.
I can't imagine how they managed to switch from one host to another. It seems almost, you know, it seems almost it seems almost counterintuitive that you would be able to get from a bird to a cow or a horse.
Yeah, it's quite interesting. So what I first saw, what I first showed you was the host species jump, and now it's in a host that is jumping actually. So it's just, especially this Chlamydia CTC is a very virulent clade, is SD24, and it can, I won't be surprised if we discover new hosts in the future.
Do you think it's actually managing to spread much more widely than people thought?
Yeah, I think so. Yeah.
That's fine. Good. Well, I'm really enjoying having a chat with Yvonne here. But has anybody else got another? So Adil's asking if all the species have a biphasic lifestyle. Or...
Yes, all the species have a biphasic lifestyle, yes. So they all have this extracellular virulent spore-like thing, which we call an elementary body. And that's the infectious particle. It's very similar like the Coxiella spore. And then that adheres and enters the cells, and then it starts unfolding its DNA. So the DNA is packed with histone-like proteins, and if one of the first genes are involved in chopping away the histone-like proteins, and then the DNA can be relaxed and can be transcribed. So they all have, and that's something which is actually characterizing the chlamydia. They have this unique biphasic developmental cycle.
They're actually amazing organisms, aren't they, in all sorts of ways. In fact, Karina's got an interesting question here. Karina, nice to see you. Is the small genome beneficial for the host switching or vice versa? It was quite interesting. You know, you'd imagine they were highly specialized with such a small genome.
I think this is a really interesting question, and actually I don't know the answer, but it's really interesting, looking worthwhile, to go in that. I think that they are all dependent on this replication inside the host cell. We still cannot culture these bacteria in some medium or whatever, so they need some components of this particular host cell, and whether particular species need other components than other species, that's not clear. We cannot figure out this when we just compare the genomes. Then we don't get this information. So I don't know, but it's an interesting question.
Yeah, great. That's great. Well, again, it would be lovely to talk on, and maybe we could think about, as I say, future events where we could go into some of these questions a little bit more. They've got one more question here, actually. Have we been able to identify some of the missing genes that make it difficult to culture on artificial media?
Well, I hope that I can discover these genes before I retire, but
I used to say things like that, but I'm not. It's coming a bit too close for me.
It's really difficult, so we don't know. We don't know yet.
Yeah. I mean, it's amazing. You'd think small genomes, you'd think you would be able to work out, but in some ways, the small genome organisms are just as hard or harder than the ones with bigger genomes to try and work out what's going on. Oh, you seem to have gone quiet, Yvonne. We can't hear you. I don't know if you've gone on mute.
Okay. I think there is a lot of information in these genomes that we are still missing, and they are actually in the intergenic regions, in the regulatory regions. That's what I believe.
Yeah. Yeah, great.
Okay.
Good. And lots of people saying thank you very much and very interesting. So thanks, Yvonne. It's so kind of you to come, and great to have you here. And thanks also for your amazing collaboration over the years. And that's great.